Welcome back to the Honor Students for these video support sessions for the course Life Science for Educators. Yesterday's session looked at microscopy, so this next series of videos is just to provide additional support on this topic. In this first video, we are going to look at the light microscope. Before we look at the actual content on the light microscope, I would just like you to listen to Dr. Nayampangendu explain the units of measurement so you can get a better idea of how small where the units actually are when it comes to looking at cells under the microscope.
So units of measurements. So the symbol for micro is this, which appears like a U, and it is a unit prefix in the system of measurements that we use, which is called the metric system, which was developed in France, and it denotes a factor of 10 to the power minus 6, and this is 10 to the power minus, which is 1 over 1 million. So what this means, micro is 1 millionth of a meter. And then milli, you know, a ruler is 30 millimeters, a meter is 100 millimeters. So milli denotes a factor of 10 to the power minus 3, which is 1 over 1,000. So 1,000th, so 1 millimeter therefore means a meter divided into 1,000 sections. Then 1 centimeter denotes a factor of minus 2. So it's 1 centimeter is 100th of a meter. And then nano is 1 billionth of a meter. What do we mean by this? Saying 1 millionth of a meter, 1,000th of a meter, 100th of a meter, or 1 billionth of a meter. So I thought I should just give you a visual of a meter. So here I measured on a whiteboard a meter. So actually I found out that the width of a whiteboard that we use in the lecture rooms, it is a width of 1 meter. And therefore, when we talk of those measurements, so if you divide this, so from here to here, it's 1 meter. And if you divide this 1 meter into 100 sections, then one section is 1 centimeter. And this we are very familiar with, because a 30 centimeter ruler, it means it is 30 sections, which each one 1 centimeter long, that we are familiar with. And then if you divide this 1 meter into 1,000 sections, and then one section is 1 millimeter. Again, we are familiar with this one, because a 30 centimeter ruler is 300 millimeters. And then if you divide this 1 meter into 1 million sections, then it's 1 micrometer. So what do we mean? I will explain now to get you to visualize how small a millimeter is, or a micrometer is. And then if you divide this 1 meter into 1 billion sections, one section will be 1 nanometer long. Right. So here, so this is one from here to here. This is 1 centimeter, and it is divided into 10 millimeters. If we just take 1 millimeter, this is 1 millimeter, and we divide it into 1,000 sections, just this one, and we divide it to say 1, 2, 3 until we get to 1,000. Each section will be 1 micrometer. That's how small a micrometer is. Dividing this one into 1000 sections, and one section will be 1 micrometer. So I thought you have an idea of how small a micrometer is.
So let's look at the light microscope now in more detail in this first video. Now the word microscope is actually broken down or comes from two words. The first is micro, which means extremely small or tiny, and usually it's too small to be seen by the naked eye. And scope means to view. So the microscope is the name given to the instrument that is used to view extremely small or micro things. And this is why I thought it would be a good idea to incorporate part of the lecture from last year that revised what was meant by a micro, so you can try and get a sense of how small these microorganisms are that we actually view under the microscope. Now, looking at the types of microscopes, there are basically two types of microscopes that are used in biology, namely the light microscope, which is what this first video focuses on, and then the electron microscopes, which will be looked at in the second video. Now, the words light and electron denote the source of energy that is used to examine the object or specimen that is being viewed under the microscope. So there are two types of light microscopes, and there are the two types of electron microscopes. So the light microscopes use light to view the specimen, and the electron microscopes use a beam of electrons to view the specimen. So the light microscope. Here is a compound light microscope. Now note that the compound light microscope has 3 objective lenses on a rotating nosepiece. Each objective lens magnifies the specimen by different amounts. Now when using, for example, the smallest objective lens might magnify the object 10 times. The second one might magnify the object 40 times. And the last one might magnify the object by 100 times. Now that with the magnification of the lens in the eyepiece or the ocular lens gives you a total magnification. So if the eye lens or the ocular lens magnifies by 10 and the first objective magnifies by 10, the total magnification would be 100 times. If it's the eyepiece and the second objective lens, so let's say the ocular lens is 10, the second objective lens is magnified by 40, that magnifies 400 times. And then with the highest objective lens, if it has a magnification of 100, so 100 times 10, it magnifies the specimen by 1,000 times its actual size. So that is the compound microscope. Here is a simple microscope. Now, it differs from the compound microscope in that it only has one objective lens. And this is the third type of microscopes. This is called the stereoscopic microscope, where you use both eyes because many students actually struggle looking at specimens with just one eye. The point of the microscope is that you use the one eye to look at the specimen, and your second eye is so that you can see what you're doing if you're making annotated diagrams of what you're viewing under the microscope. So the assumption here is that you do know what the different parts of the microscope are, and you know what their functions are. So I think this would be a good time to pause the video and for you to find a diagram of a microscope and label the parts and write the function of each part of the microscope. So let me just repeat what I'd like you to do. I found this labeled a photograph of a stereoscopic compound light microscope, and as you can see, the parts are labeled. So you can find a similar diagram, label the parts, and next to each label, you can just write an annotation that says what the function is or any other relevant information you might want to include to remind yourself about things like care for the pieces, how they can be damaged. It's really up to you as to how much detail you would like to include. So I'd just like you to watch this short video that summarizes some of the parts of the microscope and their function.
Light microscopes are useful for studying specimens ranging in size from about 1 millimeter to about 200 nanometers. It's impossible to study anything smaller by light microscopy because the shortest wavelength of visible light is about 390 nanometers, and anything smaller will not interact with the light. The most common form of light microscopy is called bright field microscopy. In this form of light microscopy, the light beam passes through the sample and into the objective lens. Let's look at the parts of a light microscope to see how a specimen is magnified and viewed. The lamp is where the light beam is initiated. This beam is divergent, spreading out as it approaches the condenser lens. The condenser lens gathers the beam of light and focuses it so that it converges on the specimen with high intensity in a small area. The specimen usually is on a glass slide that rests on a platform called the stage. When the light beam hits the sample, the light rays are either reflected or refracted or absorbed by the sample. The beam then continues to diverge upward on its path away from the specimen. After the beam leaves the specimen, it passes through the objective lens. The objective lens performs the bulk of the magnification of the specimen. The eyepiece receives the light beam from the objective lens and refocuses it to recreate the image in the viewer's eye. Lenses found within the eyepiece are called ocular lenses.
If you were alive 200 years ago, you would have been given some very strange advice on how to avoid getting ill. You might have been told to carry sweet-smelling flowers with you or avoid going to smelly places. This was because at that time, people didn't understand that infectious diseases were caused by microorganisms. And they didn't know that because they couldn't see them. Microorganisms are so called because they can only be seen using a microscope. They are far too small to be seen with the naked eye. In fact, the invention of the light microscope opens up a whole new world to scientists, the microscopic world. A microscope magnifies objects and makes a larger image. This allows us to see objects in more detail. Resolving power, or resolution, is also a feature of microscopes. Resolution is the ability to distinguish two different points as being separate. Basically, using a microscope with a high resolution means that you can increase the magnification further without the image becoming blurry. The light microscopes you may use at school will have quite a low magnification and resolution, but they will still enable you to view individual cells. To use a microscope, you place a slide containing the specimen onto the stage and secure it using the clips. You first choose a lowest objective lens by turning round the nose piece. This is usually ×10 magnification. You then turn on the light. This might be by using an electric light or moving a mirror to reflect light onto the specimen. If you look down the eyepiece lens, you will see the image, but you will probably need to use the focusing knobs to move the stage up and down until the image is clear and not blurry. You must be careful when moving the stage up not to knock into the objective lens, as this might break it. Now you can choose a higher magnification objective lens to see the specimen in more detail. You might have to readjust the focus. Now you can clearly see the specimen, you can draw it and write down the total magnification you used. This is calculated by multiplying the magnification of the eyepiece lens, which is normally ×10, by the magnification of the objective lens. You will also need to be able to use a magnification formula. Magnification equals size of image divided by size of real object. So, if the diameter of an image of a red blood cell in a book is 1 centimetre and the actual diameter of a red blood cell is 0.001 centimetres, then the magnification used must be 1 divided by 0.001 equals ×1000. The microscopic world has been opened up even further due to the invention of the electron microscope. These use beams of electrons to form images and have much higher magnifications and resolutions than light microscopes. They have enabled scientists to study objects in much more clarity and detail, from cell organelles like the nucleus and membrane to even smaller structures like individual atoms.
The first video outlined the parts of the microscope, what their functions were, and how they work together for producing an image using light. It also noted what the smallest specimen could be because of the wavelength of light. The second video by Fuse School, entitled How to Use the Microscope, showed the parts of the microscope and also how to use the microscope. And it included defining what resolution was and showed you how to calculate the magnification of the specimen being viewed under the microscope.